Assalamu alaikum everyone, my name is Muhammad Dawar Zahid and I'm a first year medical student at Aga Khan University, and welcome to the video on the Golgi body / the Golgi apparatus. Before that, we've done a video on the endoplasmic reticulum, we've done a video on the nucleus and on the mitochondria as well, so please do go and watch those if you haven't already. And let's just start with a really brief introduction of the Golgi body. You've probably read this line in your O-level books as well, that the Golgi body is the post office of the cell. And we'll talk about what that means in a minute. Additionally, structurally, it's quite similar compared to our SER, but it is more compact compared to the smooth endoplasmic reticulum. So that's it with the brief introduction. Okay, so what happens is that I want you guys to sort of try and understand this as a kitchen. So what happens is the let's just call it the oven. So we have a pizza cooking in the oven. So it's cooking over here, right? So I want you guys to think of the oven as the endoplasmic reticulum. That the pizza is being made in the oven, but before it leaves the oven and gets into the it gets brought out to your table, it has to undergo, between these two steps, it has to undergo some form of, some form of modifications. And in this example, those modifications are going to be, in the example of the pizza, it's going to be, let's say we're going to drizzle some olive oil on the pizza, or we're going to cut it, all of those things. So we need these modifications happening, and these modifications are basically the Golgi body. So what I mean by this is that in the endoplasmic reticulum, we have proteins, we have lipids, we have all of these things being made, but before they can leave / do their function, we need to process them, we need to modify them, and that is where the Golgi body comes in with the modification. And this might seem a bit repetitive to you guys, but I really need you guys to try and visualize and try and imagine what's happening, and once you do that, talking about the functions, it's going to be pretty easy. Okay, so one more thing that I want you guys to sort of, let's just put it all in front of us. So we have the smooth endoplasmic reticulum, we have the rough endoplasmic reticulum. Smooth creates lipids, and the rough one creates proteins. Now, they can either be used as is, that is to say that they don't want any further modification. The cell just decides, okay, we just want this, barely made form, let's just use it. But in most cases, we need to modify them. And that is where we have them going to the Golgi body. There are a lot of names. There's, Golgi body, Golgi apparatus, Golgi complex. And I'm saying all of them again and again because I really want you guys to familiarize yourself with all of these, terms. It goes to the Golgi body, and after going in the Golgi body, getting processed and all that, it will leave the cell in most cases. In certain cases, it might also stay, but so most of the cases, it will leave. Let's just talk about the functions of the Golgi body, and I want you guys to pause the video, make sure that you have understood the mechanism before we can really get into the functions, because if you have understood the mechanism, the functions are going to be really easy. So the first thing that we have is all of this processing that we need to do, and that is going to be glycosylation. Fancy term, but all it means is adding a carbohydrate or a carb group. It's basically a short form for carbohydrate, a carb group. If we have a lipid, that's going to be called glycolipid. So I have a lipid coming from the smooth endoplasmic reticulum, it will be made into a glycolipid once it passes into the through the Golgi body. If I have a protein coming from the rough endoplasmic reticulum, it's going to be made into a glycoprotein. Pretty simple, right? So moving on to the second thing, we have enzyme formation. So enzymes, as you guys know, are proteins, right? If you don't know that, so I just told that to you guys. So we have these proteins, they undergo modification and they're able to become enzymes. What that entire process is, we don't really care right now. Moving on from that, we have the third thing, which is going to be our lipid, I'll just call it, lipid transport modification and storage. Okay, so talking about the lipid transport, it's pretty simple. So if I have, a piece of lipid, I want to transport it from one part of the cell to the other, it's going to pass through the Golgi apparatus, Golgi body, Golgi complex, whatever you want to call it. There's also, modification, and if I want to store the lipids, it's also going to get stored in the Golgi body. So moving on to the fourth thing, which is going to be the formation of primary lysosomes, and we'll be talking about them in a bit. So I have all of these primary lysosomes, and that's going to be it. But one more thing, sorry, I forgot that, and I'm going to write that in a different color because it's really important for you guys to understand, that is labeling. And let's just go with the pizza example. So I had the oven, I had the processing desk where I'm going to cut the pizza, I'm going to put on olive oil or whatever. The oven is going to be the endoplasmic reticulum, the processing phase is going to be the Golgi body. So over here I'm going to do one more thing. If let's say that the pizza has to be delivered, I'm going to put a tag on it with the house address, or whatever I'm going to put on the receipt, the bill, whatever. I'm going to tag it for a specific place, that it has to go to the specific place. Either it has to be delivered outside the pizza shop, or it has to be delivered to a specific table. Let's say it has to go to table 12. So I'm going to tag it, and that's exactly what it means by labeling. The specific molecule which is undergoing all of this modification, it has to go someplace, and I need to tell it that okay, I want you to go to this place. And that is also why I call it the post office. So it basically tells you the address. It tells you that okay, I've created insulin, I needed to go from the pancreas to whatever the target organs are, and that is what it tells, that okay, these are your specific target organs, these are where I want you to go. Okay, so guys, let's start with the visualization of the Golgi apparatus. Before I explain the diagram, I want you guys to sort of pause the video and try and understand what's happening, try and read through the labels and everything. Okay, so I expect you guys have done that, and let's just quickly start. So over here I have my endoplasmic reticulum, whatever type it is, I don't care. And from the endoplasmic reticulum I have these incoming transport vesicles. They're going to come and they're going to pass through the Golgi apparatus, they're going to get processed, they might just move like this, they might just move like this. I don't really want to concern myself with that right now, but once they're done, they're going to come towards this other end, this other face that I have of the Golgi body, and they're going to leave the Golgi body in the form of these secretory vesicles. It might also leave in the form of lysosomes or whatever, but I'll basically call them secretory vesicles at this point. And you can see the formation of a vesicle, it's sort of pinching off over here. So let's just take the example of a protein. So let's say a protein comes from the rough endoplasmic reticulum, and that protein is now going to pass through, just a second, I'm so sorry, let's just use it like this. So that protein is going to pass through the Golgi apparatus. It's going to get let's say it gets glycosylated, it gets processed, modified, all of these things, and then it gets labeled. Let's say it has to leave outside the cell, let's say it has to go to the brain for a specific purpose, so it's going to get labeled. And once all of that is done, once we've passed through it, it's going to leave the Golgi body, and then it can go wherever it has to go. Okay, so guys, let's move on to the second organelle that we'll be talking about in this video, which is going to be the lysosomes. And the reason why I did these two organelles in one video is because they're really interlinked, and I wanted that we can sort of understand how both of these work in the same video. So these are basically single membrane bound vesicles containing hydrolytic / acidic enzymes. Basically really powerful enzymes that have the ability to eat away at material. So for example, we have proteases, which are able to break down protein, and then we have lipases, which are able to break down at lipids. And one more thing that I want you guys to know is that we can have up to 50 enzymes in one. Let's just write enzyme, but we're going to have up to 50 enzymes in one single lysosome. So do you guys see how powerful these are actually? And they're also 1 micrometer in diameter, so they're really small but really potent and really powerful. So basically we have these enzymes, and they're sort of like hitmen. They're like a hitman or like an assassin. So what they do is that they essentially kill the stuff that we want to we don't want anymore. So they're sort of like killing machines, if you may. Okay, so let's talk about the lysosome, how it actually works. And I've made this beautiful illustration for you guys. So over here we have something called our primary lysosome, and we just talked about that it's filled with all of these really powerful enzymes. So let's just color it purple, or let's just color it red. So let's say that this is something that we want to break away. We don't want this anymore. So this is let's say material we do not need. Let's say it's some old organelles or whatever it is. Let's talk about that in a second. So what happens is that it basically, the primary lysosome, it moves and it fuses. The word is fuses. It fuses with the organelle, or with whatever that we don't need anymore. So all of those enzymes that we had, they're going to sort of flow out into this vesicle. And just like I told you guys, it's going to fuse and form this thing, this other vesicle that we'll be calling a secondary lysosome. And the way I want you guys to remember this is second. Okay, two. We have two things over here. Previously we only had one thing, so it was a primary lysosome. Over here it's a secondary lysosome. And what it's going to do is that all of these enzymes that we just talked about, they're going to kill everything in this. Let's just use a cross, that's going to be simpler. It's going to kill every single thing that's present over here. And now what's going to happen is, so we have two sort of things. We have soluble material. Let's say we broke something down and it had soluble material in it. So the soluble material is just going to leave the vesicle, like I drew the arrow, and it's going to leave the vesicle. It's going to leave the secondary lysosome, and it's just going to float around in the cell, and the cell is just going to utilize it. But we have certain things that are insoluble. So this will have insoluble material now. Stuff that we can't really use. Let's say it's stuff that it's not helpful, or it's just insoluble. We just don't really need it. We need to kick it out of the cell. So the vesicle, this the secondary lysosome, it's going to move towards the cell membrane. So I've made this cell membrane, and again it's going to fuse with the cell membrane, and the remaining of the enzymes, because what I want you guys to really understand is that these are literally killing machines. I don't want any of these enzymes, these red enzymes, I don't want them near my cell. I just want to use them, and then I just want to discard them. So what I do is that I throw all of this content, insoluble stuff, some remaining enzymes, and I throw them outside the cell. What happened to this arrow? So I just throw them all outside the cell. And that is how the mechanism of lysosome action and all of this killing, it actually works. Okay, so let's talk about the functions, and I sort of want to quantify it for you guys so that it's easy. The first thing is something called autophagy, and these terms are important. Okay, so what autophagy basically means is that I have old or worn out organelles that I don't want anymore. I'm like, just take them away. So I have these old organelles. So what I do is that I break them down, and the useful things I reuse them. Useful stuff reused. So I'm able to use it without the cell really losing on important stuff. The second thing is going to be autolysis. And in autolysis, what's going to happen is that this is basically cell death. So this is essentially breakdown of cell. So if I don't need a cell anymore, I'm going to break it down, and all of the useful material is going to be utilized by the cells near me. So that's what's going to happen. One more thing that I have, which is really important from an immune, let's just write it immune purpose. So immune system. Let's just write immune system. So what essentially happens is that I have these cells called macrophages and neutrophils, and they do something called phagocytosis, which is essentially this same thing happening. However, what the cell does is that the cell takes the bacteria, and it puts the bacteria inside. Let's just say that this this vesicle has the bacteria. So what the cell is going to do, it's going to put the bacteria inside of itself, and then the primary lysosome is going to come and it's going to kill the bacteria or pathogen or whatever. So we're not going to go into a lot of detail right now. We have a whole chapter, immunity wala chapter for that. So right now I just want you guys to know how it sort of fixes in with this entire mechanism that we have. Lastly is basically release of hydrolytic enzymes to clear the surroundings. And let's sort of imagine this. Let's say I have a cell over here. It's a really crude looking cell. Let's say that I have let's just say it's a cell. It's sort of like a parasitic cell, or it's like a decomposer or whatever. And let's say that I have wood all around it. So all of this is wood. And the cell, it needs energy, it needs nutrients and all of these things, right? So what the cell is going to do is that it's going to release these small, lysosomes, and these lysosomes are going to burst, and they're going to deliver all of my hydrolytic enzymes over here, which is going to break down this wood for the cell to take it in. This is also sort of what happens in fungus, in some parasites, and this is what we do, that the cell sort of creates space around it. It chews away at this wood or whatever is around it. So we clear the surroundings, and lastly, one last thing that I want to write over here is we use the material provided. So it's not really senseless killing, it's sort of killing with a purpose, if you must. So this is it with the video for the Golgi body and the lysosome. I hope you guys enjoyed it, and if I missed anything, if there's anything that you guys would like me to focus on, add more onto, so please let me know. I would honestly love to hear that. Thank you so much, and please do like, share, subscribe, and all of those things, and please do refer me to your friends, etc. That will be really helpful for me. Thank you so much.